Right, good. Yeah, hi. I will in this hour give you an overview of like the different techniques that there are to spatially profile the transcriptome in situ. There's many methods, and there's basically no time to cover all of them. I will focus on four large groups of methods, and yeah, really my goal here is to give you a little bit of a technical insight in how these different kinds of methods generate the data that you will work with in this course. All right, with that, yeah, as you know, our tissues are very complex. They consist of many different cells, many different cell types, and these are not just randomly positioned in space, but they are like highly organized in our tissues. This is true for the examples that I show you here. So in the brain, there's a lot of structure, same as the retina and any other tissue. And even in cancer, which I always thought would just be a random bag of cells, it turns out that there is a lot of structure in these tumors as well. However, the complexity of these tissues made it very hard to analyze this and appreciate it in its full complexity. However, recently, the methods that we're talking about in this course were developed to tackle this. So there is always like in this method, there's a component of RNA detection in situ, and then some analysis, and then you can get sort of a spatial cell type map, or like maybe it's not cell-based, but it's basically measuring the organization of the transcriptome spatially. So we'll focus on four main groups, like I said, heavily focused on the methodology. We will not go into all the technical details, but I will want to give you some idea of how this data is generated. And to compare the different methods, we'll focus a little bit on the resolution, the sensitivity, number of targets, and the area that can be covered by the different techniques. All right, so there's four groups, but they split into two main differences. Like there is the microscopy-based methods and there's sequencing-based methods. So let's start with the microscopy-based method. They are all sort of based on in situ hybridization. This is a method where you hybridize a probe to a target. And this probe is usually labeled with something that you can then visualize in the microscope. There's a subdivision here. On the one hand, we have barcoded FISH. FISH stands for Fluorescent in situ hybridization, which is one branch of the microscopy-based methods. And then there is the in situ sequencing branch, which uses a different way of detecting the RNA. If we move to the sequencing side, these all work with spatial barcodes. So the RNA is tagged with a predefined spatial barcode to sort of locate it in space. And there's a subdivision in two major groups. There's a spatial sequencing, which is spatial transcriptomics is an example of this, in which the RNA moves to a spatial barcode. And then on the other hand, there is what I now call spatial tagging. And in this variant, the RNA stays in place, but the barcode moves. So sort of two flavors of the same idea. We'll start with the microscopy side and then work our way down to through the four major groups. So let's start with barcoded FISH. So these are, like I said, based on in situ hybridization, where a probe is designed that is complementary to the target. So this is where the first thing, you need to know what you're looking for in this method. So you need to know your target beforehand. You need to know this, like at least part of the sequence, so that you can design a probe that will hybridize complementary to the target. And then the probe here in black, it's labeled with some sort of thing that we can see. In the earliest iterations of this method, this was usually a radioactive isotope to see. Then it moved to chromogenic stains, and you might know sort of this purplish in situ hybridizations. And in the most recent iterations, it moved to fluorescence. So here's a example of RNA scope from ACD Bio. I think this is now a very popular method to do fluorescent in situ hybridization commercially. I guess many of you, or some of you, will have experience with this. And so in this version, the red star here is a fluorophore, or in some iterations, it's a bunch of fluorophores, like an amplified tree of fluorophores, so that you can visualize it. And yeah, in the below images, you see some examples. So in blue, it's the nuclei of the cells. Then in red, green, and white, it's different RNA molecules that are targeted here. However, you see that it's for the green on the right side, you can sort of see individual molecules. So every dot here is an individual molecule of RNA, but on the red, it's a little bit hard to see individual molecules because there's just so many. So there's another variant of this, and this is called single molecule fish, or smFISH for short. And this is a very sensitive method, although and it works like this. So you have a gene target, and then you design 40 small probes for it. They hybridize the RNA or your target, and they basically tile the whole RNA. And every probe has a single fluorophore, and when you then put that under the microscope, this is something that you see. So again, here every dot is a single molecule of RNA. The color corresponds to one of the 3 genes that are chosen here. And it's a really nice method because you can now count the individual molecules per cell and say something about the cell. So for instance, if gene 1 is a marker for a certain cell type, you might say that this cloud here, this cell here, belongs to that cell type, for instance. Downside a little bit of single molecule fish is that you need a very high power microscope, so very high magnification and also very high numerical aperture and a very sensitive camera to see that. With RNA scope, on the other hand, you can, because the signal is much brighter, because it's amplified, you can get away with a lower power microscope, which is much more convenient for imaging. However, these methods are all quite limited because they're limited by the number of distinguishable fluorophores that there are in the microscope. So if you have a regular microscope, it might be 4 different colors, 5 different colors. If you have a super good microscope, it's like 7 different colors. So that means you can distinguish 7 different genes at the same time. However, organisms typically have like 20,000 genes, which is way too complex for our microscopes to distinguish in a single staining. In the smFISH-based methods, there is a solution that's been developed over the past years, and that is the idea that you can do repeated stainings on the same sample. So you do a staining on a sample, you image that, you remove the staining, and you can basically have an empty tissue again, and then you can do another round of labeling on that tissue section. On top of that, there is another idea, and that is that you can do barcoding, and I will explain that now. So here showing that you can reprobe the same tissue again. So let's say you have many rounds. On round 1, we do single molecule fish for this RNA. Now we image it, and then after we're done imaging, we can strip the probes off. There's many ways of stripping the probes off. Very simple ways like melting them by heat or chemically. You can digest them, you can cleave them. There's many ways. And then you have an empty RNA again that you can then, for instance, label another time. And this is where the barcoding comes in. So with barcoding, you can dramatically increase the number of targets that you can identify. So in this example, we have 9 different RNA species that we want to identify. but we only have 3 different colors. So we have red, green, and blue. However, if we make a barcode, design a barcode for every RNA, we can get a unique combination of colors over different rounds. So RNA B would be labeled red in the first round and then green in the second round. So here's what that would look like. So this is our image of round 1, and we see a number of dots with different colors. Let's focus on this one. So we see that this dot is blue. And what that means is that it belongs either to RNA G, H, or Y, or I, sorry. So we cannot distinguish between them yet. So we need to do another round 2, and we see that in round 2, this molecule is again blue. So here I put the dots next to each other so that we can see the barcode. So it's blue, blue, which means it's the RNA belonging to gene I. And in this way, you can dramatically increase the number of targets. In an ideal scenario, it scales like the number of fluorophores times the number of rounds, or like to the power of the number of rounds. So in our previous example, we had 3 fluorophores to the power of 2. If you move that a bit further and you have 4 fluorophores and you do 7 rounds, you can stain 16,000 targets. And if you have 8 rounds, you can do 65,000 targets, which is more than enough to cover transcriptome. However, it's not as ideal as it was originally suggested that it would scale, and that is because of 2 reasons. One of them is optical crowding. In this image, you see individual dots, but there is also like these slightly bigger blobs, and they might be 1 molecule, there might be 2 molecules, and it's very hard to say. So with this barcoding scheme, if you would stain every RNA molecule at once, the resolution of the microscopes is not high enough to distinguish individual molecules anymore, just because there's so many RNA molecules on a small surface. So that's one problem. There's another problem. Sorry, I will move to the other problem later. Here's an example of one of the earliest methods that used the barcoding. It's called smFISH, developed in the lab of Lonkai, who also developed this idea. And in this experiment, they looked at the cortex of the mouse brain. They had 100 barcoded genes that here you see the barcodes. So you see the hybridization rounds. There's 4 rounds here, hybridization. And then here you see the different colors. So this gene would be blue, purple, green, purple over the different rounds. And that's how they could identify it. In this experiment, not all genes were barcoded. You see that there is 25 genes that were done serially. So like if you have 4 colors in the first round, you would stain 4 genes. Then in the next round, you would stain another 4, and then another 4, and another 4. This is what's called serial single molecule FISH. And this was done for the reason that I said before. These genes were quite highly expressed, and they would crowd out, or quickly cause crowding. So that's why they did them serially instead of barcoded. Yeah. So yeah, this was quite a nice demonstration of the principle, and yeah, they showed 100 barcoded genes in situ, which is already quite a lot more than the handful of targets that you could regularly do in the microscope. There's another problem with barcodes, and that is that if you do more and more rounds, you also increase the chance that you make a mistake. So errors are really, yeah, if you do longer experiments, the number of errors increase, and like you might misidentify RNA. So let's say, for instance, this dot is there in the first round, but in the second round, for some reasons, the probe don't hybridize or it doesn't reach the detection threshold or something, so it's black. So now we're stuck with a red dot, and we have no clue what it is. It might be RNA A, B, or C, but we don't know. So this is the most common issue in these barcoded experiments, that you lose dots over time. Another thing that also might happen is that like if two dots are very close to each other, so in this experiment there is a green dot very close to the original red one, we might identify it as something wrong. This is slightly less common. It's mostly the dropout. But for these errors, there was another method quite famously developed called smFISH, and in this method developed by the lab of Xiaowei Zhuang, they made an error-robust barcoding scheme. So the barcodes were not as dense as before, not that every gene was stained in every round, but there's now zeros in the barcode, and on the right you see the barcodes for every gene. So every gene gets a predefined barcode like before, but now there's zeros in there. So in round 1, we're resolving 4 bits in 4 different colors, and then in round 2, we're resolving the next 4 bits in the same 4 colors. And these barcodes are designed in such a way that they're not close to each other. And I put an example of that in the bottom. So if in the left example, if gene A is labeled with 0, 1, 0, 0, 1, so that would mean it would, bit 0 would not light up, bit 1 would light up, bit 3 would not light up, 4 not, 5 would. And if there is another gene that has a barcode that is like this, you see that these barcodes are very, very similar. So if we would miss out on or not detect this one here, we would misidentify this RNA molecule as being from gene B. And this is because these barcodes are very close to each other. They use Hamming barcodes, which is a method to make error-robust codes. It's mostly used in telecommunication, but they use this great idea to make the in situ detection of RNA a bit more robust. So what they do is they don't pick all the barcodes, but they only pick barcodes that are quite far from each other. So here it says Hamming distance 1, which means that 1 bit needs to flip to mutate 1 barcode into the other. Here is an example of a Hamming distance 4. So this means that 4 bits needs to flip to misidentify 1 barcode into the other. So this is much more robust for errors. It even gives you the opportunity to correct barcodes a little bit, or at least identify that there is an error. This makes it much more robust. Here is an example of what they have here. They have 15 rounds, and you see the different molecules lighting up, and some of them have a perfect barcode, I think in white, and in red there is molecules with an imperfect barcode, but some of them they could still identify. And on the right you see the predefined barcode for the different genes. For both of these methods, the pipeline after that is this. So you do your RNA detection, you do your barcode calling, so you see the change over time, and then you match it to your library, like your dictionary of barcodes to identify the gene. And then you typically go to cell segmentation, but Naveed will also show you tomorrow different ways of doing this, because cell segmentation is not that easy. So you segment the cells on something, on some signal, some maybe additional image that you've taken, and then you count the molecules inside each cell. And from this point on, you have a gene by cell matrix. Like I said, the cell segmentation, there's many different ways of doing this. It's highly dependent on the tissue type that you have. Some tissues are very nice for segmentation, like on the top right. So here you have the nuclei, and phalloidin stain gives you a super nice border of the, or like a nice outline of each cell that you can then use to segment. And yeah, this segmentation mask is then combined together with the in situ FISH data to assign molecules to cells. Another method is to just segment nuclei. However, it's not always possible. Like here, I put an example of our data. So these are nuclei, and on the right, you see that these nuclei might be easily segmented, although it's still hard. But there is this super dense area of cells where it's very hard to segment the nuclei, and it's even harder to segment the whole cell from this data. So cell segmentation is still a little bit an unresolved problem, although there are many alternatives and solutions for that now as well. But this is your typical pipeline for the barcoded smFISH, and also basically all the microscopy methods. So RNA detection, segmentation, and then cell counting. Here's an example of one of the latest smFISH papers. There's many nice papers that I could highlight here, but here I wanted to pick this one out. It's quite recent, and they did a very cool thing. So they did smFISH for 4,000 genes, both in the mouse and in the human cortex of the brain. And in this study, they were comparing the anatomy differences or similarities between mouse and human. What is nice for now is that they see the amount of genes that they can stain, and with this they could identify a large number of different cell types in these tissues to compare mouse and human. Here's another example of smFISH, which in its latest iteration is now called smFISH Plus. So remember, this is the method from Longhai, and in this method they increased the barcoding so that they could identify 10,000 different gene targets in situ. So here you see an example of a cell, and they zoom in on this super tiny region, and they see all the different RNA molecules there. All right. Then this is a method that we developed. One of the issues with the single molecule FISH-based method, or the barcoded FISH-based method, is that they are quite slow and they don't have a really high spatial throughput. And this is because they need to be imaged with a very high power objective lens of the microscope, and this makes that the field of view is quite small. So the spatial throughput is not so high. As you can see here, for instance, this is the coronal section of the mouse brain. I would say this is about like 7-8 millimeters to give you some sort of idea. And in this study, they took like a handful of fields of view and not covering the whole thing because it would just take too long. For that reason, we developed another method where we pull down the RNA from a tissue section onto a surface and then image that, because the large part of the imaging that you have to do in single molecule FISH is Z stacking, because the objectives have a very shallow depth of field, and to cover the whole thickness of the tissue section, you need to make many images, and this makes it very slow, meaning that you can also not do a large area. So what we do is we pull down the RNA on the surface and then use barcoded single molecule FISH to image this. But now, because the RNA is all on the surface, we don't need to make as many images in the Z direction, and it's just a few in that dimension, so we can span a larger area. So here's an example of a sagittal section of the mouse brain. You see the scale bar here, this is 1 millimeter. And previously what we could do is like a tiny portion of this, but now we can image the whole area. We'll zoom into this area here, and you see all the individual molecules alongside with the segmented nuclei that we have of this data. There's many more examples of studies using barcoded single molecule fish. I invite you to read on them, but this is a sort of brief overview of some of the methods. So now let's make a summary of the characteristics of these methods. So the resolution is diffraction limited, and this means that the resolution is limited by the optical properties of the microscopy system. This is dependent on the wavelength of the fluorophore and the numerical aperture of the objective, provided that the camera setup is okay. It will give you a resolution of between 150 and 300 nanometers, depending on which fluorophore you use. The detection efficiency is typically high for single molecule fish-based methods. When you barcode, there is a little bit of drop, so it's somewhere, yeah, between 70 and 90%, maybe even a bit higher. Yield, it's our method it's quite a bit lower because we pull down the RNA and we don't pull down everything, so we have quite a bit of loss there. But typically, the barcoded fish methods are on the higher end of the scale. The gene throughput is hundreds to thousands. 10,000 is the highest that is demonstrated. So it's quite good, but there is a note here: you need to know your targets beforehand, and you need to know their sequence beforehand. And this is usually no problem for organisms that are typically studied, like mouse, rat, human, monkeys. It's fine, but if you would want to study a organism that doesn't have a genome sequenced, or you don't really know what the transcriptome looks like, it's maybe not the most ideal method. The spatial throughput of these methods is several square millimeters to maybe a square centimeter in the case of eel, but mostly it's several square millimeter. All right, with that, let's move on to the second microscopy method, which is called in situ sequencing. There's a lot of similarities with the barcoded fish, but now we identify they are identifying RNA molecules by their sequence in situ. Most of them, or like all of them, use rolling circle amplification to amplify the target so that it's easier to visualize and to make the sequencing more robust. What they do, they design a padlock probe. That's the one in blue here. It's called a padlock because there is a gap here. And these, like the two arms, hybridize on the target. Originally, the target was cDNA. So the RNA was first reverse transcribed to cDNA. But later iterations, I will show you also, now can do that on the RNA directly. But once the padlock probe has bound to the target, they are ligated together so that you now have a circle, and then these are amplified. Because it's a circle, it will just go on, and it will generate this large ball of many copies of this padlock probe sequence, and these are used for the detection. So also important to note here is that for ISS, in situ sequencing, this is a targeted method, so you need to know your target beforehand, similar to the single molecule fish methods. There's a couple of different ways how to then read the sequence. For ISS, typically there is a barcode encoded in this part of the padlock probe, and then you can do sequencing by ligation to identify this barcode. So let's say the barcode is TGCA. You will first flush in so they have probes that hybridize here on the left side, and then they have a here, and these are random nucleotides. And if there is an A here, it's linked with a red color or red fluorophore. So if the red probes hybridize to this what they call a Rolani, this amplified padlock probe, they know that an A is present in this position. Then they melt the probe off, and then you can do another one. So here, for instance, C is labeled or labeled by green. The ends are random nucleotides. So you flush in a pool where only the C is sorry, where only the C is determined, and if it hybridizes, you know that there is a G on the barcode here. And on the right, you see an example of the barcodes for different genes. There's quite a bit of publications on this that give great biological insight. I want to highlight one here. Here they looked at different cells in the hippocampus of the mouse brain. Oh, I see that I think it's now published in Nature Methods. Sorry, I forgot to update that. But here you see, very nicely see one cell moving over the different rounds. And you see that the molecules change color. And with this, they can identify the barcode and identify the target. What is nice about these methods is that because of their amplification, they're much easier to image than single molecule fish. So in single molecule fish, the signal is typically very dim and you need high power microscopes. Here, because of the amplification, it's much easier to see your molecules, which also means that you can more easily do a large area. So here they image the whole section of a mouse brain. This is another iteration of this. It's called HypISS. It is technically not a in situ sequencing method anymore, and then you see that on the top right corner I also changed the logo from in situ sequencing to barcoded fish. But HypISS uses embases the protocol of ISS, but then uses a different detection chemistry. So like in ISS, you have the mRNA, you do RT so that you have the cDNA, then you do the padlock probe hybridization, you ligate, you do the running circle amplification, but then instead of sequencing, there is a ID, and there's another probe which is called the bridge probe, to which fluorescent detection probes hybridize, and these then encode the predefined barcode that there is for every RNA. This very nice publication where they used HypISS on the human cortex. This might not be so striking, but if you don't know, if you have never looked at human tissues are usually very hard to work with because they have a very high autofluorescence in them. So it's very hard with these methods to do any RNA staining in them because the autofluorescence just crowds out any signal that you would generate. But HypISS is highly amplified, and with that they could do RNA detection in situ in the human brain and study the spatial organization there. So that was great. Another method in this family is FISH-seq. FISH-seq is, contrary to all the methods that we talked about before, it's an untargeted method. So it also uses running circle amplification. So if I understood it correctly, they reverse transcribe the RNA into cDNA, and then the cDNA gets ligated into a circle, and then this is amplified into a RNA, and then they use, I think in the earliest iteration it was solid sequencing to sequence part of the actual sequence of the RNA. So they're not sequencing a predefined barcode, but they're sequencing the actual RNA. Yeah, so this method is untargeted, which means it targets any RNA. That also means it targets tRNAs, which are super abundant in the cell. So a lot of reads will come from tRNA, but it's a great method for to have a really unbiased sequencing of your sample. So this would also work on species where you don't have a reference genome or where you don't have reference transcriptome, although then it would be a little bit hard to sort of identify which genes you're looking at. In the most recent iteration of this, it's not exactly the same method anymore, but it's a very related method. It's called expansion sequencing. It's built on the FISH-seq like ID, except that it's introduces a lot of different steps. So we start here with our tissue, some cells. They get embedded in a gel, and then they get expanded. And this is a method called expansion microscopy, where you make use of a swellable hydrogel to physically enlarge your tissue. And this is really nice because our tissues are quite small, and microscopes have quite a hard time to resolve all cellular features, especially in neurons that have many axons and dendrites that are very small protrusions of the cells, and they're typically too small to see properly in microscopes. But if you physically enlarge them, they become much easier to image. So after the expansion, and you see now that it's much larger than the starting, you do in situ sequencing of the different RNAs here. That is what you see here. So you see the sequence. This is the raw sequence of the RNA. However, it turned out that this is not enough information to identify the RNA with. So what they need to do afterwards is that they need to extract the RNA or cDNA, and then sequence again to have a full, like a longer read, to map it to a reference transcriptome, so that they can now identify the gene that belonged to that individual spot. Yeah. So here they show the data, and what is really nice, so every dot here again is a molecule. They can now show individual RNA molecules inside these axons of the cells. With most other methods, they don't have the resolution to show this, so this is quite interesting. Yeah, you see also that there is quite a few reads of the ribosomal RNA. Sorry, I said tRNA before. Most of it is reads from ribosomal RNA, but there's also many reads from other RNA. All right. Another method in this category is called StarMap. They had a slightly different approach in that they don't do the reverse transcription step, but they do the running circle amplification directly on the mRNA, and they do this with a probe which sort of resembles a padlock probe, but now the gap is not on the RNA, but it's on the side. And then they have a snail probe, what they call a snail probe, to hybridize here to bring those two ends together, and then use ligation to make it a circle. Sorry. And then they use running circle amplification to amplify this. Now there is also in situ sequencing variants where they use that can use directly the RNA. And the reason why this is important is that the reverse transcription step is not so efficient in situ. It's not super efficient. So you lower your detection efficiency if you do this step. And with this, they claim that they can have a little bit higher detection. And then they use a similar method to in situ sequencing to decode, although they're now doing pairs of pairs of nucleotides instead of single ones. Right, so let's wrap this one up. The resolution, it can be limited by the microscope setup if you use a low power objective, but another limiting factor is the amplicon size. They are typically half a micron, 1 micron in size. So this is yeah, what determines the resolution. The detection efficiency can be super low in the case of SpaceSeq, but it can also be much higher, and it differs a little bit between the different techniques. The gene throughput is between tens and thousands. I think now it's a little bit more than 1,000. In SpaceSeq, of course, or expansion sequencing, the gene throughput is unlimited because you just do de novo sequencing and it's not targeted. But in the targeted methods, it's somewhere around this range. The spatial throughput is, for some methods, several square millimeters, but it's more towards square centimeters because you don't need such a high magnification objective to image these methods because of their amplification. All right. With that, let's move on to the other side of the spectrum, and these are the sequencing-based methods. So remember what I said, there is two differences, and the main difference is like what is moving. So in the spatial sequencing, like spatial transcriptomics, the RNA is moving, and in the other one, the barcodes are moving. So let's first look at the spatial sequencing methods. First method here, spatial transcriptomics, like I said, they used a microarray with spatial barcodes. So they have a glass slide here in blue, and these are spotted with different spots, and every spot contains a unique barcode for that location. So you see the orange spot here has I depicted here as an orange bar, but this is a specific barcode that can link it to this location. And then there is a poly T, which can hybridize to the poly A tail of mRNA. So you have your array. On top of that, you place a tissue section, and then after permeabilization, the RNA will move, will diffuse, and some of that will be caught by the slide. So here in white, you see the mRNA, and now it's hybridized to the poly T with the barcode. Then they use reverse transcription and sequencing to sequence the part of the RNA to identify it and the spatial barcode, so that you can now place the RNA molecule back into space. So usually popular method, this is an example of the first publication where they looked at the olfactory bulb of the mouse brain, and they see very nicely that these different capture spots have very distinct transcriptomes depending on where they originate from. And we know that the mouse olfactory bulb is highly structured in these different layers, and that's also what they very nicely see in this data. Of course, now this method has been very successfully commercialized by 10x, where it's called Visium, and I think this is the method that generated the most data of all of these methods that we will talk about today. There's a few variants that use this idea as well. There is two methods that use an array of barcoded beads. So instead of a microarray, which are spotted onto a glass surface, these use barcoded beads that are deposited on the surface to do basically the same thing. So you make beads with a random barcode. So each bead has a own barcode and a poly T. Then you make an array out of them, you place them on a surface in some way, and then you capture your RNA, and you sequence the barcode plus the RNA so that you can place the RNA back in space. One method using this is called Slide-Seq. There's also now Slide-Seq 2, which are very similar. These use 10 micron beads, which each have a barcode. Then they get deposited on the surface. Then they need to be imaged or sequenced first to know which bead ended up in which location on the slide. That's called this in situ indexing step. And then the tissue section is placed on top of these beads, permeabilized, the RNA moves or diffuses, some of that gets caught by the poly-Ts on the beads, and then you can sequence this. And these beads, they have a lot of similarity from single-cell RNA-seq methods, where these beads are put into a droplet together with a single cell to do this. But now these beads are placed on the surface to do spatial indexing. Yeah, they show a couple of different examples of different tissues, 3 of the brain here on the top, and then the kidney and the liver in the bottom. Yeah, you see that if you're familiar with these tissues, that the spatial coverage is not very high. Here you see the full tissue section here, and what they look at is this tiny part in the middle. But it's a very neat way of doing this. The beads are 10 micron. This is another iteration of spatial transcriptomics called high density spatial transcriptomics. Here the beads are a lot smaller. They're just 2 micron in diameter, and they are deposited in a patterned silicon wafer so that they are much denser, patterned and positioned in space. And again, these beads have a spatial barcode, and they have a poly-T to capture the mRNA. And with this, the resolution is much increased. This is a method that recently came out called Stereo-seq. Uses the same idea of a patterned microarray as spatial transcriptomics, but here they made the features so that the spots very, very small. In spatial transcriptomics, they were like 100 micron. Now they are 55 micron in diameter. In the Visium platform, they might go smaller and more dense, but here in this method, they went super dense. These spots, they are roughly 220 nanometers in diameter, and they're 500 to 750 nanometer spaced from each other, so the density is very high. And yeah, similar to the high density spatial transcriptomics, we're now at a resolution that is subcellular. So a typical cell is, just as a ballpark, is roughly 10 micron in diameter. Some are bigger, some are smaller, of course, but let's say roughly 10 micron. So this method, which has 2 micron beads, and this method, which has 220 nanometer spots, yeah, you're really subcellular, and like one spot might be under, multiple spots might be under one cell. So what they do is they make this pattern chip with all these nanoballs. These nanoballs have a spatial sequence, and this needs to be read out first. So it needs to be sequenced first before you can put the tissue on. So once they do that, they ligate the IDs, the spatial IDs, with the poly-T, and then they place the RNA on top. Again, the RNA moves, gets captured by the barcodes, and then they do sequencing. They know the RNA and they know the spatial barcode to know where the RNA comes from. And in this paper, in the first publication with this method, they make an atlas of mouse development, and as you can see, they use the full mouse embryo. You see, this is half a millimeter, and yeah, they look at all different tissues in development, which looks spectacular in my opinion. So let's wrap up this spatial sequencing part. The resolution is sort of dependent by the spot size. So what we talked about is between 220 nanometers and roughly 100 micron. But it depends also a little bit, like the RNA can diffuse, so it's a little bit hard to say that this is the resolution. but this is at least the resolution of the data that you will get. The detection efficiency can be quite low in the for Slide-Seq. It can be a little bit higher for other methods, so it's roughly between 0.1 and 20% for these methods. The gene throughput, full transcriptome, it's untargeted mostly, so that is great. And the spatial throughput is several square millimeters up to a square centimeter or around about that range. All right, let's move to the last part of the sequencing-based methods. So in the previous method, we saw that the RNA moves to the spatial barcode, and these techniques, the spatial barcode will move and hybridize to the RNA. There's two methods here I want to talk about. One is called dBET-seq. This uses microfluidics to flow through spatial barcodes, and it's quite nice. You see the chip here. There is a bunch of holes here where you deposit 50 different barcodes. And then there's a number of channels in the bottom. You can also see it here. So on the right, you will put your barcodes. And then there is, like, from every barcode, there is a channel running to the other side where it's sucked up. And here there is the channels are open, and they get placed on top of, they get sandwiched on top of a tissue section. So what happens is that the barcodes will attach to the RNA that is in one of those channels. So here you see the stripes of where these barcodes have attached to the RNA in the tissue. So this gives you one dimension, right? So now that you have 50 stripes, and you know if you would sequence this, like from which stripe it would come. But what they do is they rotate the chip afterwards, and that's also the one that you see in the bottom here, or they have a different chip, again with 50 barcodes, and now they flow them perpendicular to the first one. So that's the one that you see in green. So now you see these green columns where the second barcode has flowed, and you see these squares where the red and the green overlap, and now every one of those squares will have a unique combination of an horizontal barcode and a vertical barcode. So that's the spots that you see here. And yeah, if this is ligated to the RNA, you can sequence this, and you can place these reads back into space. The channel size depends a bit. I think the lowest width of the channel size is 10 micron, and that's the chip that you see here on the right. Here the channels are a little bit wider, and this is what is determining the resolution. But the number of, or as it is now with this method, the number of barcodes always stays the same. Might increase in the future, but yeah, this is sort of, if you have higher resolution, you cover less area. That's basically what they're trying to say. It's a very versatile method, and it's maybe much more versatile method than many of the other methods that we talked about before, because you can just tag anything you want. So here, and originally they were tagging mRNA, but you can also use antibodies with the barcode to also look at the number of proteins. And in the most latest iteration, you can also do epigenetics, especially. So here they're looking at histone modifications spatially, and they can do all these things simultaneously. So it's quite interesting platform to follow because it can combine so many modalities in the same assay. The other method in this group is called sci-Space. It is sort of related to a single cell RNA-seq method where cells are tagged and then for like super high multiplexing. So what they do is they have a slide on which they print or deposit spatial barcodes, and that are these colored, like large colored dots here. And then they sandwich this with a tissue section. Then, and you see the cross section up here. So in the colors are the spatial barcodes, and they know where these are. And then these barcodes diffuse into the tissue, and they tag the cell. So they don't tag the individual RNA molecules, but they tag the cell. And you can also see that now in blue here it's the nuclei, and now these nuclei are colored with the spatial barcode that is roughly on top. What they do afterwards is, because they image this so that they know the alignment between the spatial barcodes and the tissue, and then they do a regular single nuclei RNA-seq protocol. So they sequence all the RNA that is in the nuclei, but now they also know the spatial tag. So they know where this cell is roughly coming from. The spots are 73 micron on average in size. There is 222 micron between the spots. And after they sampled everything, they get roughly, they recover roughly 2.2% of the original nuclei that were on the slides. But yeah, this really neatly combines spatial methods with already highly optimized single nuclei RNA-seq methods. And they also here look at mouse development in its entirety. So you see here is the head, and here's the tail on the bottom. All right, with that, want to conclude the spatial tagging part. So the resolution between 10 and 100 micron, somewhere there. It depends a bit on the different methods. The sci-Space has maybe a little bit more issues with diffusion of the barcodes. With dbit-seq, there is quite a bit of signal lost in between the channels, because there is like really a physical channel pressing down on the tissue, which destroys everything in between. So yeah, that is the data that you will get. The detection efficiency is quite okay. It's between 15 and 20%. Gene throughput, full transcriptome, of course, and the spatial throughput is several square millimeters. I think in the dbit-seq is in a little bit on the lower end, and I think the sci-Space has a lot more potential to expand the area, because you just need to make a larger array with more barcodes. All right. Yeah, so we talked about the different methods, the microscopy-based method, the sequencing-based methods. There's many methods that I did not mention here. There's also other groups that I didn't mention here, but this hopefully gives you a brief overview of all the main groups. If you want to know more, there is a super good review, which is called the Museum of Spatial Transcriptomics, that discusses all spatial methods in very high detail. The article that's online, it's super dense and it's a little bit hard to read, although all information is there. But they have a super nice, sorry, they have a super nice website which contains a full-length article, full-length review, which discusses everything in super high detail. And yeah, if you're interested in these different methods, I would highly recommend you reading this, because it's a very comprehensive review of everything. All right, with that, thank you very much for your attention. Good luck with the course. And if there's any questions, I would like, please feel free to ask them.